Our DNA suffers millions of damaging events each day. Fortunately, our cells have powerful mechanisms to repair damage, and most of the time, these mechanisms remain active and functional throughout old age. However, over time, some damage will fail to be repaired and will stay in our DNA. Scientists think this damage and a decrease in the body's ability to fix itself may be an important component of aging. Most DNA damage is harmless. For example, small errors in DNA code, called mutations, are harmless. Other types of DNA damage, for example, like when a DNA strand breaks, can have more serious ramifications. Fixing a break in a DNA strand is a complex operation, and it is more likely the body will make mistakes when attempting this repair. These mistakes could shorten lifespan. Mutations occur when a mistake is made during DNA replication. About once every 10,000 to 100,000 times, DNA polymers will add the wrong nucleotide or add too many or too few bases in a specific location. Most mistakes in the nucleotide order are repaired by a cell's error correction machinery. This damage is usually repaired at cell cycle checkpoints. If the damage is irreparable, the cell self-destructs. Mistakes that do make it through the system of monitoring and repair or destruction are called mutations or alleles. A mutated gene and an allele are essentially the same. They are an alternate version of the same gene. Different alleles arise because mutations cause nucleotides to be substituted, deleted, or inserted within a gene. These different alleles are the part of our DNA that is responsible, for example, for different hair color and different eye color among siblings and parents within a family. Alleles are also responsible for the presence or absence of freckles on the face, detached or attached earlobes, and a straight hairline versus a widow's peak. If a child has blue eyes, that means both of his parents carried an allele for blue eyes. If a child has brown eyes, that could mean both parents carried an allele for brown eyes, or one parent had an allele for brown eyes while the other had an allele for blue eyes. Since brown eyes are the dominant gene, the blue-eyed allele would essentially be null. That child would have brown eyes. These types of mutations are normal for human life and are passed along to children constantly. There are other types of DNA damage that can be passed along that are harmful. Mutations in non-sex cells or somatic cells are not passed on to children. Many forms of cancer, such as lung cancer and skin cancer, result from such mutations. Mutations in sex cells, or germ cells or gametes, can be passed along to the next generation of children. And these are the damaged DNA that are harmful and that can be passed along to children. Cancer is caused by mutagens and carcinogens. Mutagens are chemical or physical agents that can damage DNA by changing its nucleotide sequence. Carcinogens are mutagens that cause cancer by damaging DNA. Another kind of DNA damage buildup occurs when a cell divides and passes its genetic information onto its two daughter cells. During cell division, the telomere, a stretch of DNA at each end of a chromosome that doesn't encode any proteins but instead protects the protein encoding part of the DNA, becomes shorter. When the telomere becomes too short, it can no longer protect the cell's DNA, leaving the cell at risk for serious damage. In most cells, telomere length cannot be restored. Extreme telomere shortening triggers an SOS response, and the cell will do one of three things. 1. Stop replicating by turning itself off, becoming what is known as senescent. 2. Stop replicating by dying, called apoptosis. Or 3. Continue to divide, becoming abnormal and potentially dangerous. These are the cells that can lead to cancer. Cancer is unrestrained and uncontrolled cell growth and division. Cancer can lead to large masses of cells called malignant tumors that can cause serious health problems. The cells in these tumors begin to redirect crucial parts of the body, i.e. blood vessels and oxygen, to that tumor rather than the organs they were supposed to go to. This is what causes the sometimes often deadly consequences of cancer. Treatments for cancer focus on killing or slowing the division of cells using chemotherapy and/or radiation. Down syndrome is another disease caused by damaged DNA. In every cell in the human body, there is a nucleus, where genetic material is stored in genes. Genes carry the codes responsible for all of our inherited traits and are grouped along rod-like structures called chromosomes. Typically, the nucleus of each cell contains 23 pairs of chromosomes, or 46 total, half of which are inherited from each parent. Down syndrome occurs when an individual has a full or partial extra copy of chromosome 21. This additional genetic material alters the course of development and causes the characteristics associated with Down syndrome. There are 3 types of Down syndrome. The first, called trisomy 21, or non-disjunction, is when Down syndrome is caused by an error in cell division called non-disjunction. This results in an embryo with 3 copies of chromosome 21 instead of the usual 2. Prior to or at conception, a pair of 21st chromosomes in either the sperm or the egg fails to separate. As the embryo develops, the extra chromosome is replicated in every cell of the body. This type of Down syndrome, which accounts for 95% of cases, is called trisomy 21. Mosaicism occurs when non-disjunction of chromosome 21 takes place in one, but not all, of the initial cell divisions after fertilization. When this occurs, there is a mixture of 2 types of cells, some containing the usual 46 chromosomes and other containing 47. Those cells with 47 chromosomes contain an extra chromosome 21. Mosaicism accounts for about 1% of all the cases of Down syndrome. Research has indicated that individuals with mosaic Down syndrome may have fewer characteristics of Down syndrome than those with other types of Down syndrome. The third type is translocation. This accounts for about 4% of all cases of Down syndrome. In translocation, part of chromosome 21 breaks off during cell division and attaches to another chromosome, typically chromosome 14. While the total number of chromosomes in cells remain 46, the presence of an extra part of sickle cell anemia is another type of disease caused by DNA damage. Sickle cell anemia is an inherited form of anemia, which is a condition in which there aren't enough healthy red blood cells to carry adequate oxygen throughout your body. Normally, your red blood cells are flexible and round, moving easily through your blood vessels. In sickle cell anemia, the red blood cells become rigid and sticky and are shaped like sickles or crescent moons. These irregularly shaped cells can get stuck in small blood vessels, which can slow or block blood flow and oxygen to parts of the body. Sickle cell anemia is caused by a mutation in the gene that tells your body to make hemoglobin, the red, iron-rich compound that gives blood its red color. Hemoglobin allows red blood cells to carry oxygen from your lungs to all parts of your body. In sickle cell anemia, the abnormal hemoglobin causes red blood cells to become rigid, sticky, and mishappen. The sickle cell gene is passed from generation to generation in a pattern of inheritance called autosomal recessive inheritance. This means that both the mother and the father must pass on the defective form of the gene to the child for him to be affected. If only one parent passes the sickle cell gene to the child, that child will have the sickle cell trait.